Hello. When I'm late with the class, let me know and I will stop it. I sort of went over there. Any questions? And start the screen share. All right, let's get rid of that. So what we're doing today is Lab 2, Aseptic Transfer. Today in the lab, you'll learn how to successfully transfer from one container to another and doing it aseptically. And you will be doing it virtually. So please pay attention and watch the videos and all that. Let me state that you will need to read the textbook in chapter 3 through the end of the section on compound light microscopy. So you need to read that before starting labs 3 and 4 next week. Oh, wait a minute. You'll need to do that before Thursday because you're starting lab 3 on Thursday. All right, any questions about that? Let me remind you that this week you can take the practice quiz by Tuesday. And if you do take it, besides giving yourself a good practice on a practice quiz, meaning how to take a quiz, I'll give you 1 extra credit point if you do take that practice quiz. And then the quiz, the real quiz for the lecture in the lab over chapters 1 and 3 and lab 00, meaning the lab safety, and lab 1 will be due by Friday, 11:59 p.m. on Friday. And you must take it from midnight on Thursday night to any time on Friday to 11:59. After that, the quiz will no longer be available. Any question about that? Any question about anything? So this week we'll be doing labs 2 and 3. You'll be doing lab 3 on Thursday, but lab 2, excuse me, not Thursday. You'll be doing it on Wednesday and lab 2 today. There's no questions about that. I'm going to minimize that and I can get rid of that. We don't really need that. Let's go ahead and talk about the lab. So lab 2 is on aseptic transfer. The primary learning objectives: complete this laboratory. Students should be able to understand the purpose of using aseptic transfer techniques, determine if aseptic transfer technique was used from the data provided, and define the following terms: ubiquitous, aseptic technique, inoculation, contaminant, and sterile. You will be using online resources to watch some videos. I need to move that over there so I can scroll down. Bacteria and microorganisms are ubiquitous. When we say they are ubiquitous, it generally means that they are essentially everywhere. They're in the air, on our skin, on the lab benches, on every surface you can think of. Let's try putting that over there. This is a problem when you're working in the microbiology lab, because when we generally are working, we want to work with pure cultures. And a pure culture is a culture that contains one and only one species of microorganism. And if you're doing work, you don't want to contaminate your pure cultures. The concept of pure cultures came up in the mid-1800s by Louis Pasteur, who developed a technique for doing aseptic transfers to minimize the risk of culture contamination. And he also came up with the concept of a pure culture, but his procedures for obtaining a pure culture were really difficult. And so you will not be practicing his procedure to make a pure culture. But anyways, when you're working and you're going to be working virtually in the lab, you do need to use the technique of making a aseptic transfer to minimize the contamination of your pure culture. And contamination is the accidental inoculation of unwanted organisms into your pure culture or the sterile media. And generally, when we're inoculating things, even virtually in the lab, we'll be giving you sterile media to do the inoculation. Another advantage of using aseptic technique is when it's used appropriately and correctly, you will reduce the possible risk of contaminating your environment and making you or anyone else in the lab sick. And so we're going to learn about how to make aseptic transfers today. So follow the lab safety protocols like wear closed-toed shoes in your virtual lab, button your lab coat, use gloves, tie your long hair back, etc. Work on your workbench and you should disinfect it before you begin work and then after you're done. And you guys just need to do it virtually. When you're inoculating things, never lay the test tube caps or petri dishes on the workbench. Hold them. Your inoculating loop should never be left down unless you're going to re-sterilize it. The petri dishes, you should hold them while you're using them, and then when you're done with it, put it back on top of the dish. There's some other things you can do to minimize the contamination, like when you're getting into a tube, hold the tube at an angle because things generally fall down straight into the tube. And if you hold it at an angle, you have less chance of something falling in the tube. If it's a solid tube, you can actually hold it at a 90-degree angle, and then something getting into it from the air would be extremely remote. Of course, if it's a liquid tube, you can't hold it at a 90-degree angle. When you're working with a plate, you can hold the lid over the plate, and that will reduce the risk of something falling down from the air, and it'll hit the lid of the plate instead of falling into the plate. When you're doing your work, and even when you're doing your work virtually, try and work efficiently. Don't talk while you're making transfers. Sterilize your tools like the loops or the needles beforehand, and then after you're done. Before you leave the lab, you should disinfect the workstation, remove your gloves and then your lab coats, and then wash your hands. And that's always a good technique when you're practicing aseptic technique and should always be done. So learn the terms. I'm not going to go over them. Here are some of the tools you use to inoculate, and that means to transfer cells into a container, inoculate a container. We won't use the flame, even virtually. We will use a Bacto incinerator to sterilize your wire loop. I think you're only using a loop today. Whenever you see references, if you have links to them, you don't have to work or click on these links. These links aren't even clickable. It's done well. So in laboratory 2 exercises, you're not physically going to performing these activities, but you will be watching the videos and doing some of the activities virtually below. We expect that you will know and understand the materials in these videos and in the activities you'll do. In procedure 1, you're going to transfer from a sterile liquid using a glass serological pipette, which is not covered in the video, but there's some reading for it. Transfer in procedure 2, a sterile paper disc using forceps, which is not in the video, but I think you've all used forceps before. So you just clean them beforehand. You can't really sterilize forceps, but you can clean them by heating them up, not so hot that you can't hold the forceps, but clean them and then pick up the sterile paper disc and put it into the tubes. In procedure 3, you'll be transferring Serratia marcens to a broth, which is also not covered in the video. Oh, wait a minute. The second one was covered in the video. All right, I read that wrong. Sorry. Procedure 1 is covered in the video. Procedure 2 is covered in the video. Procedure 3, you're going to transfer Serratia marcens to a broth using a loop. It's not covered in the video, but you sterilize the tube, and there's a little bit that's covered in the video by putting it into a back-to incinerator, and then you cool it, and then you pick up from, let's say you're picking up from your stock container, take off the lid, put in your wire portion into the container and put it into your sterile medium. So that's procedure 3. And there's an activity you'll be doing in the lab with procedure 3. And then procedure 4, transferring Serratia marcens to an auger slant. Also not covered in the video, but you will be working with a loop in the slant. And I thought you were transferring from a petri dish to another dish in the slant, which is very similar to what you're doing in procedure 4, except you'll be doing it into a slant instead of into a petri dish. Serratia marcens is a bacteria which is colored pink or reddish, and that will color the inoculum or the media once you transfer it in and it grows. So go ahead and watch your video. Watch this video and then do procedure 1. I'll have you read through that. I'm not going to talk about any of that. You must use appropriately labeling the tube, and you should label it with your initials and the date. And this isn't correct. You should also put on the type of media if you know it, and it's nutrient broth. And then optionally, you can put the, well, you should put the inoculum, what you're inoculating it with, as well as you might put the temperature. That's optional. Let me state that when you're reading something, a meniscus, like in a barrette or in a pipette, the water will stick to the sides, and so it won't, the water won't go across straight. And how you read that is you read at the bottom of the meniscus. For some reason, that's not showing the 1.0 mil. So you read that at the bottom of the meniscus. You do not read it at the top of the meniscus or in the middle of the meniscus. You read it at the bottom of the meniscus. And I guess you'll be working with a serological pipette today. In lab 2, use an aseptic technique to transfer a sterile paper disc into the nutrient broth. And since it's a sterile paper disc into sterile nutrient broth, if the technique works, you will get no growth in the tube. And I see why this is red. This should be corrected. And that is, you should label the nutrient broth with your initials, the date, the type of media, which is nutrient broth, and what you're inoculating it with. And this is just the paper disc. Okay. So if it's done correctly, you shouldn't see any growth, any turbidity in the tube. If it is not done correctly, you will get contamination in the tube and you'll see turbidity. In procedure 3, you'll use a aseptic technique to transfer Serratia marcens into a sterile broth tube here. It's a liquid growth when we call it a broth culture, so it's a liquid media. And the cells will grow at a very high density That makes it difficult to determine if there's been contamination in the tube, especially if there's minor contamination, because the cells will all grow together. So if there's minor contamination, you can't determine it in a broth culture. This one is labeled correctly. Use a small piece of labeling tape to make a label for your broth culture. You should add your name, the date, the type of medium, in this case, NBYE, nutrient broth with yeast extract. You can put the incubation temperature, that's optional, but you should put the inoculation. In this case, you're inoculating the tube with Serratia marcescens. And I'll try and make the corrections there and post that if you don't have the corrections on your webpage. Procedure 4, you're going to transfer Serratia marcescens from a a stock culture to a sterile slant culture. The slant culture is shown here, and you put bacteria on the slant by using S-type motion on the slant. You don't stab into the auger. You're just inoculating the slant. If it grows, you'll see red bacteria growing on the slant. You can get high cell densities there, and the advantage of the slant is because it has such a small opening and a large amount of media, this can last, the cells can last for a month or maybe even 3 months if they're refrigerated. And so the slants can last longer than a plate. A plate is good for a week. You might be able to get 2 weeks out of a plate. All right, that's procedure 4. Any questions about that? When you're done with those procedures, then you will see the results. Those results are given to you in this lab module 2. And here you're seeing P1 and P2. If you have any questions as to what is what, just ask me. P3 and P4. So P4, of course, is the last procedure for here, inoculating the slant. And then the results are given to you, read through the results, and then answer the questions. And you can't really type in any answer here into the lab module, but I've got a worksheet. Let me show you where that is. Bring that over here. Click on the class, and then scroll down to lab 2, week 2. And there's the worksheet. So open that up, and then you can fill out the worksheet. You have to click on that and download it. And then you can type in your answers. And then you should have a button over here for downloading your worksheet. Download it in this folder here, and then I will grade your Lab 2 worksheet. If you have any questions, I will be on the, staying on the, what do you call this, the Zoom until 3:45, okay? So if you have any questions, I'll be here to answer them. If you don't see me, it means I've stepped away from my desk, but I'll be returning within a minute or two, okay? Any questions? If there's no questions, go ahead and get started on the lab, and I'll be here to answer any questions. I'm going to stop the share now and stop the recording.